Okay, so when we left off, we were talking about introns and exons and how your introns are the things that are inserted between the coding sequence of your DNA. So those things have to be processed out. So you'll make an mRNA transcript, and then you have to cut out the parts that are non-coding sequences and splice together the pieces that are coding sequences. So you could imagine that this is a very sensitive process because if you had a DNA sequence, okay, and I have to cut out, introns are bigger than 2 bases, but let's just say that that's what I need to cut out is 2 bases, okay, or 2 nucleotides here. I got to cut out those 2 nucleotides. If I accidentally cut out 3 nucleotides, then all of a sudden I have changed the entire coding sequence of that mRNA. Okay, so it codes the coding sequence is every 3 nucleotides. Okay, so like if this was the start of the coding sequence, it would be CGG, and then what I should have is I splice out AA and I go CCC, TTT. Okay. But if I make a mistake and I cut out AAC, now that changes the entire coding sequence from CGG to CCT, TT, whatever's next. Okay, do you see how it shifted it? And now it spells out something different than what it should. You ruin the entire protein. So you can see how important it is that you cut out the exact intron. And nothing more, nothing less. Because if you leave some or you cut out more than you're supposed to, that's going to cause what we call a frame shift. And a frame shift will result in a nonsensical protein. So you'll make a protein that just makes no sense. Okay? It doesn't do any jobs because it was just created incorrectly. All right? So RNA splicing must be very precise. It has to be, or else it's just not going to work. Okay. The spliceosome is the enzyme, the protein enzyme that helps with this process. It is responsible for making sure that you cut out the piece that's necessary and to splice together the exons, which will result in the coding sequence of the mRNA. Okay, so here we have some exons in blue, okay, and in red we have the introns. So we got to cut out the introns. So what happens is that you can see this intron is smaller than this intron. That one's bigger, that second one. So you can see what it does is it loops the intron together, and then it cuts it right there at the junction, and it splices together, the spliceosome splices together the 2 exons at that junction. Okay, so it's going to kind of loop it and then splice it right there. That's what it's going to do. So that spliceosome is going to do that process. Okay. Okay. So the genetic code is referred to as the code of the nucleotides that result in a specific amino acid being put in a spot of a protein. Okay, so that genetic code codes for the amino acids. There are 20 amino acids. There are 4 nucleotides. Okay, so we have taken a language, a nucleotide language, and we have encoded it into a amino acid language. Okay? All right, so the genetic code relies on 3 base or 3 nucleotide codons, what we call codon, that is going to code for a specific amino acid. Okay? So more than one codon can code for the same amino acid. So maybe CCC and CCG code for the exact same thing. Proline. they happen to code for proline. Okay, so both of them. In fact, there's more than just two that code for proline. Sometimes you have multiple codons that code for one amino acid. Okay, there's a reason for that. So none of the bases are shared between consecutive codons. No non-coding bases appear in base sequences. Okay, so this is very interesting. All organisms use the exact same code. From the simplest bacteria to the most complex human, we all use the exact same code. So if I want to express a protein in bacteria, I'm going to use the genetic code that I use to express a protein in humans. Okay? Which just gives further evidence that there was a beginning and that code was conserved throughout evolution, right? So we can have, I guess we can just see that there's a lot of evidence that that is where it started. That you start out with something simple, it works, and you keep it. It's selected for, so you don't get rid of it. Okay. So when something works, you just keep it. Could you use a whole different code? Sure. Could use a different code. I don't have to put proline on the CCC tRNA. I could have put proline on the ACG tRNA. So why did I put it on CCC? Because that's how it's been ever since the beginning. Okay? So all organisms use the same code. Okay. Messing with the vents. Okay. So 64 codons, 61 code for amino acids, and the other 3 are stop codons. So what happens is there's no tRNA for those 3 codons. So if I have that code in my ribosome, which is the thing that does the translation, there's no tRNA that's going to bind there to that codon. Why? Because instead of a tRNA, you're going to bind a release factor. Okay? And that's why we refer to it as a stop codon. Because when that release factor binds, it finishes translation releasing the peptide, amino acid peptide, and it breaks open the ribosome and stops translation. Okay, so we'll look at that more closely. So when you have an amino acid a nucleotide sequence that codes for the same amino acid, it usually has 2 bases conserved, the first 2, and then the last one is the one that changes. Okay? So CUU, CUC, CUA, CUG all code for leucine. That is an amino acid. Leucine is an amino acid. Okay? We've drawn it. And if I have one of those 4 codons, I will have a leucine amino acid at that position. Okay? Now, why would I want to only change the last of the 3? Well, because that helps ensure that mutations won't affect my outcome, okay, of my protein. This is called a silent mutation. So let's say that this nucleotide got changed from a U to a C. It doesn't matter. It doesn't affect anything because I still have the exact same amino acid at that position as I would have had anyways. So it's called a silent mutation. There's no effect to the protein. And that's what we care about. Remember, DNA to RNA to protein. Protein's the final outcome. Once I make that protein, it has a job to do. If it's made correctly, it's going to do its job. So whether I have this codon, CUU, or CUC, it doesn't matter because the outcome is exactly the same. So having that last nucleotide code for, if you change it, it still codes for the same amino acid, just builds in resistance to mutation. Okay? Now, if you change the first of those nucleotides, one of the first ones, then you're in trouble. Okay? Then you are going to get a mutation that could potentially cause a drastic effect to your entire protein, and that might disable it and make it inactive. Okay? All right. So here is the genetic code. Okay, so the genetic code allows us to translate. It allows us to translate an mRNA sequence. So if I had the mRNA sequence ACC, actually I'm going to start with ACG. ACG. Why isn't my pen writing? That's why. Okay, start over. ACG, GC, oh, beautiful. C, AUU. Okay? So if this was my sequence, my coding sequence, I have 3 codons here. So I'm going to add 3 amino acids to this peptide. Okay? So I find A, I find C, and then I find G. Okay? ACG. threonine. So at that position, ACG, I'm going to add a threonine amino acid. Okay? Next, GCC. GCC. Alanine. So next will be an alanine. AUU. Isoleucine. Okay? So there I have my tripeptide. Okay? That's my tripeptide coded from the base sequence of the mRNA. Okay? So that's how we would use this genetic code. You are going to have to use the genetic code. I would provide it for you, and you're going to have to translate an mRNA. Okay? So you're going to have to be able to use it and know how to use it. But it's not very difficult. You just find the codon sequence, and it tells you which amino acid is present. Okay? So this tRNA would code for asparagine. Okay? AAC, and it would have an anticodon of UUG. Okay, that's how they're going to find each other, because the mRNA sequence is AAC, the tRNA sequence is UUG, and they're going to match up. They're going to pair, and because there is an asparagine attached to that tRNA, that's the next one that's going to get added to my peptide. Okay, that's how this process is going to work. Okay. All right. We don't need to go through that again. We would find the amino acid from the genetic code. Okay, so protein synthesis. We call it translation. because your mRNA is nucleotide language, your protein is amino acid language. You got to change the language, so we're going to translate it. Okay? So translation is carried out on ribosomes. Ribosomes are complexes made mostly from rRNA, and then they have also large amounts of proteins embedded into the rRNA. Okay? But it's mostly RNA with some protein embedded into it. The proteins are stabilizing that RNA enzyme, this ribosome enzyme. And the ribosomes are some of the biggest molecules in your entire cell. You can actually see them. You can actually see the ribosomes when you look through a microscope, because they're the dots. When you see dots in the cell, those are the ribosomes. And also on the endoplasmic reticulum, you have a smooth ER and you have a rough ER. The rough ER is rough because it has ribosomes on it. Okay? And those ribosomes are doing what? Making what? They're making proteins. Okay? That's why there's so many ribosomes, because they're making all the proteins that are doing all the jobs in your cell. Okay? All right. So protein synthesis occurs in multiple places on one mRNA at a time. Okay, so you can start translating an mRNA, and as that ribosome moves forward, the next one can jump on, and then the next one can jump on, and you can just start translating it at multiple places. Or if there's more than one binding sequence on a single mRNA transcript, then you can make 3 different proteins from one mRNA. Okay, that's also possible. Okay, so that's a polysome, which is an mRNA plus the multiple ribosomes would be a polysome. tRNAs, they, as we know, bind to specific amino acids, and their anticodon will recognize the appropriate codon on the mRNA. Okay, so here's a schematic, simple schematic of this translation process. This is your DNA. It is double-stranded. Okay? You are going to make an mRNA template from only one of the strands of the DNA. So the templates, so here is the coding strand. The coding strand is going to be the same sequence as the mRNA. The template strand will be the one that gets copied. Okay? So here you can see I make a copy of the template. T to A, G to C, A to U, C to G, G to C, G to C, etc. G to C, okay? T to A to U. I'm making a copy of the DNA sequence. And once I've done that, that mRNA, if it's bacteria, is just going to right there and then start to be translated. Okay? It's right there in the cytoplasm. Boom. It's ready to go. If it's inside of a nucleus, like in eukaryotes, then it has to get the guanosine, you got to get a phosphorylated guanosine cap and a poly A tail. Okay? You got to splice out the introns, put together the exons, so that packaging has to occur, and then it gets shipped out through the nuclear pores. There's these little holes in your nucleus that the mRNAs shoot out of, and then they're in the cytoplasm. And once they're in the cytoplasm, bunch of ribosomes there, they start to get translated. Okay? All right. So now this mRNA is ready to be translated. And it does so by the tRNAs recognizing the right sequence. So here is an mRNA. It's on the ribosome. Okay, so my ribosome has a big subunit and a small subunit. The mRNA kind of fits between them, and the ribosome clamps onto the mRNA. Okay, so whatever coding sequence is in this first site, which we say is the A site, okay, so if I have a CCC there, then if a tRNA comes down, that has, let's say, AAA anticodon, it's going to come in here. It's not going to be recognized, but it's not going to bind to the CCC, and it's going to leave. Okay? So those tRNAs are just going to keep going in there until the right one binds with the right amino acid, and then it's going to get added to the peptide chain. Okay? So that's how this process of translation is going to occur. You have to have a good concentration of all of the possible tRNAs and amino acids present just in the cytoplasm, okay, at the right concentrations, and then they're going to go try to get in there, and the right one's going to find that anticodon-codon interaction, and then it's going to be accepted, okay? And once it is, then the amino acid of that tRNA is going to be positioned so that the tRNA before it, which has the peptide attached to it that has been growing, okay, this amino acid is going to get added there, and when it does, the bond down here between the amino acid and the tRNA is going to be broken, releasing this amino acid from this tRNA, okay, and then that tRNA is going to leave. And then everything's going to shift over. And now this tRNA in the first site is going to move to the second site, and then it'll have the entire peptide chain attached to it. And then the next amino acid is going to come in on the tRNA, on the next tRNA. Okay? You're going to create that peptide amide bond, carboxylic acid, amine. React those together, you form an amide, right? So amino acids have a carboxylic acid on one end, they have an amine on the other end, and so when you react those together, they form an amide bond, a peptide amide bond. Okay? And when that bond forms, it breaks the bond at the tRNA, releasing the tRNA, and it shifts over. And then the process can continue and continue until there's a stop codon reached, and that stop codon says, stop translating, right? Because the release factor comes in instead of a tRNA. There are no tRNAs that are going to match at that codon sequence. Instead, there's an actual protein that recognizes that codon sequence. And it recognizes it, it goes in, it breaks the bond between the tRNA and the peptide, releasing the peptide, and then it breaks open the ribosome. Remember, the ribosome's two pieces. So it breaks apart the ribosome, and then everything starts over. Okay? So that's what that stop codon is going to do. Okay, so the ribosome are complexes of RNA and protein. Each ribosome is made of two subunits, a large subunit and a small subunit. The small ribosomal subunit contains 1 RNA and 33 proteins. The large ribosomal subunit contains 3 rRNAs, ribosomal RNAs, and about 49 proteins. But those rRNAs, even though there's just 3 of them, are ginormous. They're big, okay, and they fold up, and they fold up to the point where they can be active enzymes. So they have to fold up correctly. And they do, all on their own. Okay? Okay. Many ribosomes on one mRNA make a polysome. All right, so here we can kind of see these different subunits. Here's the large subunit. Here's the small subunit. Okay? When those two come together, then they form a functional ribosome. Okay, but first the mRNA is going to bind to the small subunit, and then the large subunit kind of clamps on top of that. Okay, that's how that's going to work. They kind of show it kind of being more of like a sandwiching it, right? In this, so you can think of it either way. That large subunit, I like to think of it, the large subunit coming down and clamping on to the small subunit on top. Okay, so then it's going to be bound to an mRNA. The ribosome is going to move along the mRNA, and as it does, tRNAs are going to come in. They're going to add their amino acid. The deacylated tRNAs, meaning the ones without the amino acids, are going to leave, and then everything's going to shift over. And you're just going to keep doing that process, and that's translation. Okay? And then you're going to make your peptide sequence, which will then fold alpha helices, beta pleated sheets, and then those alpha helices and beta pleated sheets are going to fold into a functional protein. And now you have a functional protein. Okay, so tRNA's job then is to make sure the correct amino acid is added at the correct part of the protein. Okay? So each tRNA has a covalent bond to a specific amino acid at the very top of it. Okay. The enzyme aminoacyl tRNA synthetase is what covalently links the proper amino acid to the proper tRNA. Okay. So here you have a protein. Down at the bottom here, it has an anticodon recognition site. This particular aminoacyl tRNA synthetase will only recognize one anticodon. And so when that anticodon is recognized down here, okay, that's the tRNA that's going to fit. Okay, and up here is the only amino acid that's going to fit onto that protein. And so when you get the perfect fit of both things, they come in, and then the tRNA, the synthetase will link, covalently bond the amino acid onto that tRNA. Okay, so that's how we can ensure that the correct thing is getting added right there to the top. Okay. Okay. Now this tRNA is released. It's ready to go off and do its job. Okay. So it's just fascinating how many processes have to occur for any one process to actually occur in our body, right? Yes.
So the actual
That's the synthetase. It's an enzyme, protein enzyme. And it has binding sites that are specific for only very specific things. Okay, so only this amino acid will fit there. Whatever that amino acid is, alanine, glycine, isoleucine, whatever it is, they're all going to have their own synthetase. And they're going to bind, and that synthetase will only accept one amino acid. That way it can ensure it links the correct tRNA to the correct amino acid. Okay? All right, so when you are going to perform translation, it starts out with an initiation process. Okay? So, which require initiation factors. These initiation factors are proteins. They're going to be bound to the mRNA or to the ribosome, and they're going to position everything correctly. Because where you position the mRNA on the ribosome will ensure that the correct protein is translated. Because if you don't, so for example, here's my small subunit, here's my large subunit. Okay? Here's my mRNA. AUGCCA. Okay? So if this was my coding sequence, AUGCCA, if that's the sequence that is going to make the correct protein, then I have to make sure that this codon is the first one that gets translated. But if I shift it over accidentally just a little bit, now UGC is the first codon. And then CCA is the next one. I have just ruined my entire protein. Okay? It has to be positioned into the correct reading frame. What we call a reading frame. If you shift the reading frame, then it is nonsense. Okay? So this reads the cat. But if I shift it, now it's nonsense, right? If I shift the reading frame, now it makes no sense. So it's a nonsensical read. And the same thing happens on your proteins. If the correct reading frame is positioned, then it will produce the correct protein. But if you're shifted at all, one way or the other, if you mess with the coding sequence at all, it will lead to a nonsense protein being made. Okay? Which won't be able to do the job that it's intended for. So this process has to happen correctly, which is why we have these initiation factors to line up the mRNA correctly onto the ribosome so that the first codon that is read is the start codon. There is a start codon. Okay? And that start codon is AUG, and it codes for methionine. The very first amino acid on every single protein is methionine. That is the first codon for every single reading frame. That's how it knows where to start. It finds that AUG and it positions it right at the start. Okay, there's all this other nucleotide sequence before it, and it just skips over that, finds that AUG, and puts it right in the start codon sequence, or start site, that A site. And then that first methionine tRNA comes in and starts the process of protein synthesis. Okay? Okay. After initiation, you get elongation. So elongation, we've kind of described a little bit. The tRNA binds to the A site of the ribosome. And so you kind of have the A site, the P site. Some people like to think of an E site, which is the exit site. You don't always have to think of that one. A and P, the peptidyl site, the aminoacyl site, and then this P site is where you're going to get that peptide bond formed, and then the tRNA is going to be released. Okay, the A site is going to be where it accepts the tRNA, and then allows for the new amino acid to be added to the peptide chain. Okay, so you're going to form a peptide bond. There is a protein that helps catalyze that reaction. Okay? So everything has to be positioned perfectly and in place for this reaction to occur. If I take tRNAs and I just throw them into a test tube, those amino acids on the tRNAs are not going to just start, forming peptides. It's not going to happen, okay? There's too much tRNA around it, and it's just not going to happen, okay? So, on the ribosome, though, you position everything perfectly in place so that those two tRNAs are perfectly aligned, so they will come together, and then a protein will help bring that bond, that peptide bond formation, okay, to occur. So that's how this process is going to happen, with the help of a lot of stuff around it, like a ribosome and this peptidyltransferase, okay, which is going to cause that peptide bond formation. Then their translocation is where you're going to shift everything from AMP over, and then the tRNA is going to leave, and now you have a new tRNA in the P site, and so now the A site's empty, so the next tRNA will come in and bind to the correct anticodon-codon interaction, which will position the next amino acid so that peptidyltransferase will create the peptide bond, and then it shifts. Okay? A lot of processes use ATP to do that kind of stuff. This one doesn't. It uses GTP instead. Guanosine triphosphate instead of adenosine triphosphate. Okay. So you're going to get that translocation of the ribosome down the mRNA chain until you reach that stop codon. That's elongation. So here's an example of initiation. Look, here is the start codon, AUG. It's on the small subunit of the ribosome. It's positioned. It's in place. Okay? It's in the, so they have this kind of backwards. It's the A site. So this is the A site. This is the P site over here, where the peptide bond will form. Okay? So this tRNA comes into the A site. It has the correct anticodon. That means that this is methionine. Methionine is the first amino acid on every protein made. Okay? It comes in, it finds that, and it then shifts over. It shifts over from the A site to the P site. That's all weird. Okay. Oh, well, first off, this is the small subunit. We need to get the large subunit attached. So we're going to have the large subunit now come in and attach. And that will start this process, this tRNA process. That's initiation. Okay? Now, once it's begun, now I have a free A site. My P site has this elongating peptide in it. Okay? This tRNA comes in, matches with the anticodon, and then it's going to, peptidyltransferase is going to create the bond between those two, releasing this green tRNA from the peptide, breaking the bond between the peptide and the tRNA, releasing that tRNA, and then everything's going to shift over. And then it'll continue, and then it'll continue, and then it'll continue. That's elongation. It will continue until you reach a stop codon, which will signal for termination. Okay? Termination is upon finding a stop codon that allows for the A site to be empty. There's nothing that's going to bind to the stop codon except for the release factor. Okay? That release factor is going to bind to that A site codon, and that's going to signal that this process needs to be terminated. And then that termination factor is going to break the bond between the tRNA and the peptide, releasing the peptide and the tRNA, and the whole thing's going to fall apart. The ribosome is just going to break apart. Okay? And everything's over. Okay. So here you see the release factor coming in. And it's going to cause this bond to break, releasing the peptide, and then everything just gets broken open. The ribosome just breaks open, and everything is just now free. Okay? It's free. All right, so let's talk a little bit about mutations. Mutations to your DNA. So mutations are mistakes that are introduced into the DNA. What if a mutation is introduced into an mRNA? So the DNA is okay, but let's say you were translating or transcribing the DNA into mRNA and you made the mRNA incorrectly. Okay? No big deal. All right? Sorry. It's not a big deal. You're going to make 100 other copies of that mRNA. One of them gets mutated, whatever. You're going to make plenty of proteins that work, and then the ones that don't work just going to be nonsense, and they're just going to get degraded, and no big deal. So when does it matter? It matters when the mutation is in your DNA. That's when the mutation matters. It doesn't matter if the mutation is in an mRNA. It doesn't matter if the mutation is in the one protein gets made bad. You're going to make 100 others, and they're going to be fine. Okay? So it all starts at the DNA. And if the mutation's in the DNA, that's when it's going to affect all 100 plus mRNAs that are made and all the proteins that are made from those mRNAs are going to be affected if it's in the DNA. So the DNA is the sensitive portion to mutation. If mutation happens in the DNA, that's when it's bad. Now, if it happens in one cell, it's fine, whatever. Apoptosis, that cell dies. You're done. It's done. It's out of there. You have millions of other cells that are fine. Okay? It happens at birth, or I should say at conception. That's when the mutation is going to really make a difference. You inherited the mutation, that's going to make a difference. Okay? That's when you're going to have a genetic disorder that can be expressed inside of you. All right? So why do we care about radiation and those sorts of things mutating our DNA if it doesn't really matter? Because it mutates one cell and whoop-de-doo, that cell's dead and nobody cares. Because it's just going to die and it's not going to do anything. When it matters is when you disrupt the cell cycle. When a mutation from radiation disrupts the cell cycle, that's when it matters. Because all of a sudden, that cell is growing uncontrolled. It has no more regulation around its growth. And it starts to become a mass of cells. Okay? And it's forming a cancerous mass. That's when we care. Okay? So you guys go walk out in the sun and mutate your cells all the time. Okay? Some of you probably sit in the sun and sunbathe and whatever, and you're okay. But when you're not okay is when one of those mutations, of all the thousands of mutations that you've had throughout your lifetime, when one of those mutations results in a cell cycle disruption and the cell starts to grow uncontrolled, that's when it matters. Okay? Otherwise, we have all sorts of protection against mutations. Okay? So, many mutations have negative effects on the health of an organism. Mutagens are also carcinogens and cause cancer. So something that is carcinogenic will cause cancer with multiple exposures. Okay? Like we said, if it's a carcinogen, that means it has the potential to mutate DNA. Does that mean you are going to get cancer? No. It means that there's a likelihood you could get cancer if you're exposed to that thing. So, working at the dental office or in a hospital, you're exposed to radiation all the time. Okay? But the precaution is that you try to limit the exposure, because you're going to be exposed to certain amounts of radiation, and that's not going to be so bad, but the more often you're exposed, the higher the chances that the mutation that you're going to acquire from that exposure will lead to a cancerous event. Okay? So I get exposed 10 times and 10 of my cells die. I get exposed 1,000 times and 999 of those cells went through apoptosis and died, and they're gone. One of them becomes cancerous. Okay? That's when we care. All right. Okay, so let's look at the different types of mutations that can occur. Okay? Point mutations. Point mutations are when only a single base is changed in your DNA sequence. So here, my original or wild type, I don't know why they call it a wild type. That's what they call it. The original coding sequence is the wild type sequence. Okay? And the mutated sequence is the non-wild type sequence where the mutation shows up. So here's my wild type, ATG, GAC. Down here it's ATG, CAC. Okay? So a point mutation could be silent if that point mutation resulted in the expression of the same amino acid as was there before. In this case, it won't, because the first nucleotide instead of the third nucleotide of the codon got mutated. So that's going to result in an entirely different amino acid. Now, does that mean the entire protein is just worthless? Maybe not. A single point mutation that causes one change in the protein, it might not make that big of a difference. So I put a different amino acid at that position than another one. It might not matter. It might not change anything. It might only reduce the activity of that protein. Maybe it doesn't destroy the activity, it only changes the activity, lowers it. So it still works, just not as well as it used to. Or it disrupts the entire protein and it's non-functional. So individuals who have a certain type of Lou Gehrig disease, ALS, can acquire just a single point mutation which changes one single amino acid in the protein that causes the disorder. Okay? So they can be sensitive. It depends on which amino acid you change. Some amino acids, no big deal. Other amino acids that you change, yes, big deal. Okay? So you inherit this point mutation, and now you have a familiar form of ALS that will result in you acquiring that disorder later on in your life. Okay? Deletions. So deletions can result in frame shifting. So if you remove an amino acid, a nucleotide from the reading sequence, then that shifts the entire reading frame, and now you have a nonsensical portion of your protein from that point on, wherever that deletion occurred. So if you delete one amino acid, you'll get a frame shift. 2 amino acids, you'll get a frame shift. You delete 3, then you just deleted an amino acid at a specific position, but the rest of it is fine. Okay? So maybe that's a big deal, maybe it's not. All right? But if you cause a single nucleotide frame shift or a 2 nucleotide frame shift, that's pretty much always a big deal, because everything after that is just nonsense. Okay? So deletions and insertions, which is the next one, can have huge effects on the entire protein if you cause only a single insertion or a double insertion. Triple insertion, you just put a different amino acid at that position, but everything else is fine. Okay? So maybe a big deal, maybe not. Frame shifts are almost always a big deal. You don't want to ever really frame shift something. Okay, so yes, go ahead.
I was just going to say, my friend's baby has deletion mutation.
Oh yeah?
They don't know, I guess it's really rare what she has. She might not be able to talk or walk ever.
Yeah, I mean, it can really make a big difference.
She's only one.
Yeah. Well, I mean, it started from, I mean, that's where it started, right? She probably wasn't exposed to any radiation or anything like that. I mean, so she inherited that disorder, or at some point during some time when she was undergoing these processes. So in our bodies, we have processes of diversifying our genes between parent and children, and so the genes can kind of jump around and move sometimes. And during those processes, you can get some mutations and things too. So it could have happened, sometime early on in the process, and now she's inherited that disorder, which is sad. So, and that's what we encounter in life. I mean, the number of different disorders and diseases out there is just astounding. And it all happens early on, right? Okay. UV damage. So UV light, ultraviolet light, can damage your DNA, and it causes covalent linkages between pyrimidine bases. So if you have a T and a C right next to each other, those pyrimidine bases, it might cross-link them, which would make it so that you wouldn't be able to transcribe that portion, or it would be transcribed incorrectly. It wouldn't be recognized what it's supposed to transcribe to. Okay, so it might insert just a random nucleotide instead of the correct one at that position. So the pyrimidine dimer on a DNA strand, this can actually be a method to kill bacteria, right? So if we turn on UV lights under these hoods or something, then that can effectively sterilize stuff. So oftentimes, like with Petri dishes, you might buy Petri dishes and it says sterilized through UV radiation, and that's all it means, right? They irradiated it with UV light, and that caused cross-linking at the DNA. Yeah? Absolutely. That's what it's for. Killing microbes inside the water. Yeah, absolutely. That's what that would be doing. Okay. All right. Recombinant DNA restriction enzymes are enzymes that cut DNA and that put DNA back together. Okay. Well, restriction enzymes are the ones that cut it. Ligase is the one that puts it back together. Okay. So recombinant DNA is interesting because it has this cool way of recognizing specific sequences within DNA and cutting at those spots. Okay. It has to be double-stranded. It'll be double-stranded DNA, and you get this what's called a palindrome sequence. Okay. Okay. So here is an example of this type of sequence. Here's a restriction enzyme called EcoR1, okay, coming from E. coli. This restriction enzyme was found in E. coli. That's why they call it EcoR1. And so this EcoR1 enzyme will find on DNA that it is exposed to the sequence GAATTC. GAATTC. And notice that the top sequence is exactly as the bottom sequence read backwards. So restriction enzymes, that's what we call the palindromic sequence. Recognize these palindromic sequences, and they will cut right between the red spot here on both. And it makes a stagger cut. And that's really important, having a stagger cut like that, because if I want to put something back together, having that staggered position will allow me to do so. Because it will, if I cut this open at that spot, then this section of the DNA will open up. But if I wanted to put it back together, then this A and this T would come and they bind to this A and this T, and they could sit there, and then you put a ligase in, and ligase shuts it back together. So you can open up the DNA, and you can close it back up. Okay? This is something that we do oftentimes in the laboratory. So if I want to make insulin, insulin is a protein. It's a human protein. Bacteria don't have it. But guess what? Bacteria use the same genetic code as humans do. So if I put a DNA sequence into bacteria, they will express my human protein. So I take a circular piece of DNA, I cut it open with EcoR1, and then I take another piece of DNA that I cut on both ends with EcoR1, and now the EcoR1 pieces of each side will recognize one another, and I can ligate it, and then over here I can ligate it, and now what I've done is I've taken this piece right here and I've inserted it into this circular piece of DNA, and then I take that circular piece of DNA and I put it into bacteria. It's a funny word that they call this. They say you transform them. You transform the bacteria by putting this new piece of DNA into them. Okay? So you think of like a transformer, but it's nothing like that. Now they have a new piece of DNA in them. Okay? So now they have a new piece of DNA in them, and they will express that DNA. Unregulated. They'll just express tons of it. If you put the right promoters and everything on there, it's going to be a protein factory. Those bacteria, they're just going to express tons of it, and then you harvest the bacteria, and you basically bust them open and get all their guts, and you purify that protein, that specific protein. And now I have insulin that I can distribute to individuals who need it, right? So that's how we can express human proteins in bacteria, through recombinant DNA processes. They also refer to this as genetic engineering. You can genetically engineer things so that they will express certain proteins that do specific jobs in places where they might not have been before. Okay? And a lot of times that happens by putting DNA that's non-native to that cell into it. Okay? But first you have to create that DNA. So you can cut up DNA and you can make DNA strands. If I want to take a DNA from a jellyfish, I can isolate the DNA from a jellyfish and put it into a rabbit and make it glow green. Okay, I can do that. Because if I know the coding sequence of that protein, I can amplify it into billions of pieces of DNA that's just that coding sequence for just that protein. And then I can take it and I can put it into another organism and express it. And now they're glowing green, that organism. Okay? That's called genetic engineering. Or if there's a plant that doesn't survive, like rice, very well in one environment, and so people are dying in some third world country because they can't grow rice, then you can genetically engineer that rice, change it so that it will have certain characteristics that make it resistant to the harshness of that environment and allow it to grow there. And now they can grow their rice. They can harvest it and eat it. And they can survive and they can be alive, right? So there's a lot of ways that this is good, but people are afraid of it too because they're like, oh, one day we're going to make super babies, superhumans. Yeah, they do this with animals all the time, right? Chickens that are genetically engineered, bulls that are genetically engineered so that they're like super muscular. So, genetic engineering is not a new thing, but it's something that can be done. But through recombinant DNA processes, by being able to cut pieces out of one place and put it into another place. Okay, using restriction enzymes and ligase, okay, we can modify the genes inside of organisms. So here are some common restriction enzymes: BAMH1, ND3, SAL1, Beagle, and PST1. Okay, so these are some common restriction enzymes, and you'll notice that they're all palindromes. So if I read it backwards, GGATCC. Okay, it's the same forwards and backwards. That's how they are. They're always like that. So that's how they are in all organisms. I mean, it's just amazing that those sorts of things are just conserved throughout, always. In fact, the use of these restriction enzymes was very instrumental in the process of sequencing the human genome. So they took a human individual and they decided to sequence that person's entire genome. And so now there is a library of this person's DNA that you can go and look at, their DNA sequence of their entire 23 chromosome sequence, right? And this was called the Human Genome Project, which was thought to be almost impossible until they started doing methods that made it very usable, plausible, I should say. Okay. All right. So here, this is what I was talking about, right? So you can take the circular DNA. Circular DNA is called a plasmid. This is also called cloning. You've heard of cloning, right? But you often think of cloning as like, I'm going to make a replica of myself. Okay? I mean, that can be a cloning process, but you can also clone just a single DNA sequence, a single protein. I can clone a single protein, and I can clone it, and then I can put it into another organism, and they will express it. Okay? That's called cloning also. So cloning doesn't have to be an entire organism. It can be just a single DNA sequence. And so when we clone things, we cut open plasmids, we amplify DNA, and we put it onto the plasmid, and then we can transform that into bacteria, and they will express it, or into another organism. Okay? And that circular piece of DNA is called our cloning vector. Okay? Because once that vector gets into that plasmid, gets into the bacteria, they're going to make like hundreds of copies of it. There's just a bunch of plasmids all over that bacteria. They're just going to replicate it. Because you put an origin of replication on the plasmid, and then the bacteria are just going to replicate the plasmid, and then they're going to make the protein that's on every single copy of that plasmid that's inside of that bacteria. And if you transform it into, thousands of bacteria, and then those bacteria grow into a colony of bacteria, hundreds of thousands of bacteria, then you're producing, billions of copies of that protein inside of that colony of bacteria. And then you harvest it if you want, or you study it, or whatever you're going to do with it. Okay? All right, so here, let's see if this will render better. Here you can see, here's the beta globin gene. So here's a strand of DNA that contains that beta globin gene. And I want to get it into this plasmid. So I cut this plasmid open with the same restriction enzyme that I cut this piece of DNA up with. And then there is a piece where that contains the beta globin gene, and I can insert it into that plasmid. Are these other ones going to insert? Yeah, they're going to insert too. But then so you're going to put this into a bunch of plasmids, like hundreds of plasmids, and maybe 20 of them have this and all the other ones have these, okay? And then you put that DNA into the bacteria, you transform it, and then you grow them individually, and you find the one that has this. And you just pick that. I pick that one because it has the gene I want. And all these other ones I just throw away. Okay? We're so mean to bacteria. I mean, just ruthless. Okay? But nobody cares. So it's when you get into higher organisms that, people care. But with bacteria, there's no activists. So you can just do whatever you want to them. Okay? You just throw them away. And you just pick the one you want, right? And now it has my gene. And there it is. And then I grow that one bacteria. I grow it up into a colony of bacteria. Hundreds of thousands of bacteria that are all expressing this. Okay? Okay. All right. So, yeah, genetic engineering. So this can be thought of as genetic engineering. Don't worry about that. Okay. All right. Let's go ahead and take a break before we get too much more. I just marked you present
Okay.
just now.